Okay, I'm ready when you are.
Okay. So, um, so this is the module two review session. Um, I guess I'll go ahead and get into it. There's 10 questions on the study guide, but we divided it. So I did five and then Hennis did um,
Fine.
the other five. So I'm gonna go ahead and start with question one. Um, so for the articles, I we obviously didn't go into depths about annotating them. So when I took cognitive psychology with Dr. Sims, she um, recommended that we take note of the introduction or the main research question, which is just like in the method, how the study was conducted, the participants, the procedure, stuff like that, the results or the statistical findings, like numbers, stuff like that, meaning for future research and stuff like that. And I actually had a student ask me this, but if the article does not include, if it's not like about a research study or it doesn't exactly follow this format, if it's like missing something, um, don't worry about it. I would just recommend reading through it carefully and basically just taking note of the important findings, the main idea, things like that. She just, in my experience, she likes to include these articles because they connect to the lecture material. Um, but yeah, moving on to question two. Um, do you mind scrolling down a little bit?
This is the question. And here's everything.
Okay, I can't read the question, but um, oh,
I'll pull it. That's the question.
okay, cool. Let me read the question real quick, and then you can scroll past. It's a little hard since she's sharing the screen. But okay, so question two is what can research about genetic defects, chromosomal defects, and teratogens tell us about the nature of development? Does research in this area impact the definition of development? Why or why not? And then explain and give some examples type and epigenetics. You can scroll down. Thank you. Defects and teratogens reveal that development is impacted by not only biology but also the environment. So it's kind of like nature nurture. It's like a combination of the two, not just one or the other. Research in this area changes the definition of development to not only include physical growth, but how the genotypes or the genetic makeup are expressed in the phenotypes or like the physical appearance. I'll go into the examples in a second, but these can be impacted by environmental triggers, including teratogens. That's like alcohol, drugs, pretty much anything negative to impact development. Um, but some of the terms she included, they're pretty basic terms. You've probably seen them before. First are genes or units of DNA carrying instructions for traits. And chromosome are structures in cells holding genes. Humans have 46 or 23 pairs of them, so they come in twos. Genotype is the genetic makeup. An example of that is having the gene for blue eyes. And then the phenotype is physically expressing the genotype, which is physically having blue eyes. Um, and then epigenetics is how environmental factors can influence genes without changing DNA, but impacting environment or development, my bad. And then on to question three. Um, she had said describe a genetic disorder, but I went ahead and just put all the genetic disorders down there and just briefly describe them. The question says, describe the genetic disorders we discussed, explain dominant and recessive inheritance patterns. What does it mean to be a carrier? So first, a carrier is a person who has one copy of a mutated gene for a recessive genetic disorder, but does not show any symptoms, but they have a chance to pass it on to their children. Like I said, I went ahead and just like briefly described all of the genetic disorders. First is Huntington's disease. This has an autosomal dominant inheritance pattern. It's the progressive breakdown of neural cells, and it usually causes a variety of problems, ultimately resulting in death. It usually occurs like in people get diagnosed in like their 30s and 40s and usually die a couple years after being diagnosed. But only one copy is needed to develop the disorder. That is cool.
All right.
And then sickle cell anemia has an autosomal recessive pattern for inheritance. It must have two genes for the anemia, one from mom and then one from dad. It affects the types of blood cells that you have. So instead of shaped more round, it's shaped like a sickle or like a crescent moon. Sickle cell anemia has two genes, and then sickle cell trait just has the one gene. So usually it's a lot more manageable, doesn't show a lot of symptoms. But having the two genes or copies results in sickle cell anemia, and it's a lot more severe. And then PKU, oh, someone else joined. Hello. Um, PKU, this is found in like Diet Coke and Splenda and stuff. Um, Having too much of it, it's, well, first, it's an autosomal recessive pattern, but when too much of it accumulates in your brain, it causes problems like brain damage. It's usually pretty severe, and it requires two defective copies to show. And then Tay-Sachs disease is also an autosomal recessive pattern. It destroys nerve cells in the brain and spinal cord during infancy. I believe it causes, actually, never mind. That's a different disorder. But both parents must be carriers for a child to develop this. And it's usually pretty severe. I think I remember her saying that basically no infant survives. Um, then muscular dystrophy, I think. It's an X-linked recessive inheritance pattern. It's the progressive weakness and eventual loss of muscle function. Usually males with the mutation are affected, and females can be carriers with just mild symptoms, and it can be generated by one carrier instead of two. And then spina bifida, I think. I'm sorry if I'm not pronouncing these right. This is a multifactorial inheritance pattern. I believe it's like either or a combination. This is the neural tube defect leading to physical and cognitive disabilities. I believe it includes paralysis of the spine. And it has been declining because of folic acid. I believe women are taking folic acid during their pregnancy. So this has caused like a significant decrease in finding this disorder. And then lastly, retinoblastoma. That is an autosomal recessive inheritance pattern. It's a rare eye cancer affecting young children, causing vision loss or eye removal. If they find this in children, they usually have to remove the eye, but it's just cancer in the retinas. This can be hereditary or sporadic, either one, and it only needs one copy needed. And the next question 4, explain the following terms regarding inheritance: trisomy, mosaicism, and chimera, I think. What are the 3 main examples of trisomy? So first, defining trisomy, that is 3 copies of a chromosome, usually from non-disjunction during meiosis. This leads to developmental delays, physical differences, and organ issues. There's 3 main types of trisomy. There's trisomy 21, which is also known as Down syndrome. That can be shown, I believe she said, in like the normal form, and there's also a mosaic form of Down syndrome. Trisomy 18 or Edwards syndrome, and trisomy 13, which is Patau syndrome, I think. Both of those are pretty severe compared to Down syndrome and have a lot more like of graphic effects. I also believe she mentioned trisomy 16, but those are the three main examples. Then going into mosaicism, this is two or more genetically different cell lines within the body occurring after fertilization. This means there's milder or partial forms of genetic disorders. So, like, you can have a mosaic form of Down syndrome and not have as severe symptoms or traits. And then lastly, chimera is two sets of DNA from different zygotes, usually from absorbed twins. So, like one baby absorbing another twin, so resulting in two sets of DNA. Um, this usually results in mixed genetic features and often goes undetected without like genetic testing. And then lastly, on my side, number 5, what are the typical sex chromosome patterns for males and females? Explain the following variations: Turner syndrome, Klinefelter syndrome, XYY syndrome, and XXX syndrome. Explain why males are more susceptible to X chromosome problems. What are androgen and recessive males and guavdoches, I believe. How could disorders involving sex chromosomes affect our definitions of sex and gender? So first, the typical sex chromosome patterns is an XX for females and an XY for males. Pretty basic. And then Turner syndrome is XO, or a deletion, where there's only one X chromosome. This results in a human being biologically female with only secondary sex characteristics. So they're not fertile, they're sterile, and then they also have heart issues. Klinefelter syndrome, or XXY, which there's an extra X chromosome. This results in a human being biologically male, but they have reduced testosterone, they're sterile, and there's often breast development. XYY syndrome, there's an extra Y chromosome. This results in a human being biologically male. They're usually taller and they can have mild learning disabilities, lower IQ, but they're fertile. I believe they just have low quality sperm, so it's a little harder to get fertile. But anyways, XXX syndrome is an extra X chromosome. They're biologically female, and there's no super major symptoms except that they're taller. And going back to how males are more susceptible to the chromosomal disorders is that males only have one X chromosome, when females have two. So they have no second X chromosome to compensate if the X has a defective gene. And then in this case, females often are able to mask those defects because they have the extra X chromosome. Sorry. Androgen insensitive males, these are genetically male, so they have the XY chromosome, but they do not respond to male hormones or androgens, and they often develop female traits. And I believe they also usually identify as female. And then the last term, it's a delayed testosterone activation, or it's like the dropping of their genitals. So they're born with female parts, but they end up developing male characteristics at puberty, which is pretty crazy. And she mainly talked about this happening in the Dominican Republic. And then lastly, biological sex isn't strictly binary. It involves a lot of aspects, including chromosomal, hormonal, and physical traits. It it challenges simply referring to people as male and female, and supports that gender is more complex than that, that it has more of a complex mix than just the chromosomes. Has a lot of like societal, cultural, whatever effects. So, yep.
Okay. I'll start right into it. The question number 6 is explain the logic of twin and adoption studies. What are monozygotic and dizygotic twins, what percentage of genes are shared by the different groups using twin studies, and be able to work up through some samples of data. So twin and adoption studies are made so researchers can separate the effects of genetics and environment, and the common debate that we always have about nature versus nurture. Monozygotic twins are identical twins. In research, they're rated as a 1 because they share 100% of the genes and they result from a single fertilized egg that then splits. And then dizygotic twins, they're fraternal twins, they're rated as a half because they result from two separate eggs that are fertilized by two different sperm and they share 50% of the genes just like a sibling. And this, I put this chart right here so you can see the proportion of genes or how they're rated by like the monozygotic twins, the two sibling, dizygotic twin, mom, dad would be a half, and then all of the other ones. Then um we have what is Stern's rubber band hypothesis. This is like it's like the name says, it's about rubber bands and how they come in different sizes and how much you can stretch them and how easy they or hard they can be to break. So Stern and others use this idea about the relationship between genetics and the environment, and he suggested that genetics set the potential range, like how much rubber band can stretch, and then the environment will determine where the individual will fall between that range. And there's also the concept of resilience, as how some people rubber bands may be able to stretch more or less without getting damaged, while others may not be able to. And then an example would be how a child with lower potential raised in an stimulating environment might outperform a child that has high potential uh, raised in a deprived environment, but only up to their stretch limit. Then question number 8 is what are the stages of prenatal development? What are some of the accomplishments of each stage? What are mutagens, teratogens, the effects of some of them, and why is it so difficult to study them? What are some limitations of this research, and how can these limitations be addressed in research? And the effects of parental age. So the stages of prenatal development are germinal stage, embryonic stage, and fetal stage. So the germinal is the first 2 weeks. This is when we have conception and the fertilized eggs that travels from the fallopian tubes to the uterus, and then it begins to divide 24 hours after conception. And half of the zygotes they don't make it, and then it ends when it's finally implanted in the uterus. Then we have embryonic stage, which is 3 to 8 weeks. This is when most of the organ development happens. We have the neural tube forming, eye parts, the heart starts to beat, and um, then the fetus is very susceptible to whatever's happening in the womb. And then we have finally the fetal stage, which is from 8 to 40 weeks. And this is when we have more rapid growth, brain development, sensory development, and all of that. Pregnancy lasts 40 weeks, not 9 months. 9 months would be about 36 weeks. And then we have mutagens are those that affect the DNA. This can be inherited or acquired, such as radiation. And then a teratogen is a external factor that causes abnormalities in the womb by creating an unusual atmosphere in the womb. So some of this can be alcohol, drugs, some medicines. Some alcohol is one of them, and it can cause like fetal alcohol spectrum disorders, facial abnormalities, cognitive delays. Then nicotine could like cause low birth weight, respiratory issues, increased risk of sudden infant death syndrome. And then they added the thalidomide because it's one of the ones that Dr. Sims was talking about, which is it causes a lot of limb deformities, and the babies that were affected by this. They were called flipper babies. And now if you want to take this medicine while you're like um pregnant, you can take it while pregnant, and for you to be able to take it, they have to like you have to have a pregnancy test and all this. So it was a really historical thing that happened. Then for studying this in research, the thing is you can't have any control groups. This is why it's hard to study it. And then a lot of the peoples that are using one teratogen, let's say like they're using alcohol, they might be also using drugs, they're using multiple ones. And then there's also the lifestyle. Those they're more likely to be poor nutrition, stress. So you have all these confounding variables that um you can know which one is the one that is actually affecting the individual. And then there's also ethical constraints because you can randomly assign some people to have the exposure and some do not. And then how to address it, some scientists use animals such as mice so they can understand the effects of humans. And there's also longitudinal studies where they follow them over time. And then researchers use larger samples so they can control for those confounding variables like income or diet, so they can isolate the effects of the teratogen. Then we have different birth classifications. What are some of the characteristics of preemies? Describe some of the characteristics of newborns and what's neoteny. So we have micropreemies, which are less than 26 weeks of gestation. They weigh less than 3 pounds. Some of them eat only an ounce a day, and they spend a few weeks to a few months so they can get out of the NICU. Then we have preemies, 26 to 37 weeks. They weigh about 3 to 5 pounds. They're prone to upset stomach and indigestion because they have problems with being able to suck. So they have a hard time eating. And some of them are ready to go home in 1 to 2 weeks. And then we have a full-term baby, which is 39 plus week of gestation. They weigh 5.5 to 9 pounds, and they eat 8 to 12 times a day. And they're usually ready to go home the next day or 2 days after. There's also late-term preemies, which are 35 weeks, and like I said, they have problems with eating. So these are some of the characteristics of the newborns. So they have swelling, they have the cone head, they're very hairy, they can have rashes on their faces, they can have mila, which is like white dots on their nose, and they can have blue hands and feet. Sometimes they're yellowish. So it's just important I added this chart so you guys can see all of the ones that could they could have. And then neoteny is those traits that make us cute as babies. When they have a large head, when they have a large forehead, large eyes, full lips, a big smile, chubby cheeks. And these are important because it may evoke more caregiving behavior from adults. Dr. Wes Sims mentioned that whenever children have most of these traits, then people tend to care more for them or look at them more. So it's important. And then average size for newborns is 7.5 pounds and 20 inches. And then the APGAR test, and what does it practice? It was developed by Virginia APGAR and is given at 1 minute after birth and then at 5 minutes. And it's very conservative. We need to see improvements at the 5 minute. And then usually it's rated and 7 and above is good. And if it's rated as 7, then they need to see improvement on that number. And whatever number is rated first at the 5 minutes, it needs to improve. This is what it is. So you can either get a score of 0, 1, or 2. So we have appearance. If it's all blue, then they get a 0 score. If it's only blue at the extremities, then I get a 1. And then if there's no blue coloration, then they get a 2. Then we have pulse, we have no pulse, 100 beats per minute, and then more than 100 beats per minute. Then we have grimace, no response to stimulation, grimace or feeble cry when stimulated, And then sneezing, coughing, pulling away when stimulated. This is good. And then activity, we have no movement, some movement, and very active movement. And then last, we have respiration, which is no breathing, or weak and slow, or a strong cry, which is perfect. And yeah, that's it. We're done for today. Unless there's any questions. I know I went a bit fast. Lana, do you have any questions or we're good? Okay. Well, if anyone has any questions, we can, you can always reach out and then we'll try our best to respond as soon as possible. Okay, I'll stop sharing.
Thank you.
Thank you, everyone. Bye.